Okay, good evening, everyone. I see we have a few people already logged on. I'd like to welcome you to the National Ocean Sciences Bowl Professional Development Webinar Series. My name is Melissa, and I work at the National Office for NOSB in Washington, D.C. And I'm just going to give a quick, brief introduction, and then we'll turn it over to our speakers for the evening. I'm sure many of you who are participating tonight know that the National Ocean Sciences Bowl is an academic competition for high school students. These students are incredibly intelligent and smart and dedicated, and they get tested in the knowledge of marine and freshwater science. The webinar series, like we're holding tonight, is something that we do specifically for the coaches for these NOSB teams. However, we do allow all educators who are interested in learning about the topic of the webinar to participate, in the hopes that both the coaches and other educators will infuse what they learn from these webinars into their classrooms. This year, our theme for the competition is the Great Lakes, a window into freshwater science. So the students will be getting tested on a greater knowledge of freshwater sciences and the Great Lakes, and therefore our webinars this year are also tied to the theme. Just to give you a little bit of an agenda for this evening, we're going to have a 1-hour presentation, and then there will be a 30-minute question and answer session. So if you look on the Adobe Connect screen, you'll see that there is what's called a pod. It's the Q&A pod. You have the ability at any time while you're watching the presentation to type a question into that pod, and I will see it. So what we'll do is we'll have our presentation for an hour, and then when it's time for the question and answer session, I'll read the questions aloud to our presenters for the evening so that they can answer them for you. So like I said, at any time, type your questions into the Q&A pod. If it is something that relates to directly what they're saying at the moment, or you have an issue with hearing or anything, type that question into me as well, and I may stop the presentation quickly to ask them the question or to speak up, but otherwise we will hold the questions for the end. Just a reminder, we have all of our participants, our guests tonight, on mute, and that's why we're having you type in the question and answers. And we'll get through as many of the questions as we can in that 30-minute Q&A session. Also, in that Q&A box, at the end of the presentation, please feel free to give us some feedback. We love to know what you thought of the presentation. You can also send us an email at nosb@oceanleadership.org if you'd like to give us a comment, but you can certainly type it in that box. And if anything goes wrong with your connection to Adobe Connect tonight, the link that you used to access the meeting tonight, you can just log yourself out and log back on, and hopefully that'll clear things up. Okay, so for this evening's webinar, I'd like to introduce our two presenters. We have Dr. Carmen Aguilar Diaz and Dr. Russell Cuhel. They're both with the Great Lakes Water Institute with the School of Freshwater Sciences at the University of Wisconsin-Milwaukee. The University of Wisconsin-Milwaukee is actually our host for the NOSB finals this year, which is another reason why we have this great theme on the Great Lakes for this year. And we have quite a few people from the university participating in these webinars, which is fantastic. So Dr. Aguilar and Cuhel have agreed to do 2 presentations in this webinar series. Tonight, they are going to be discussing sort of an introduction to the Lake Michigan food web and be discussing invasive species and how they are affecting that food web. They will also be speaking next Thursday evening, and I hope that if you're participating tonight, you will also participate next Thursday. Next Thursday, they're going to be presenting a paper of theirs, putting this food web that they discussed tonight sort of into practice. So what you'll see on our website is that there is a link under their names for their webinars. There is a link to this paper that they are going to be discussing. So we hope that you will read it between this week and next week and have read it in preparation for their webinar next Thursday. So with that, I am going to turn this over to our speakers this evening, and they will start their presentation. And again, thank you for participating tonight. Okay, it's all yours, you guys.
Okay, welcome to Building an Aquatic Food Web from the Bottom Up. I am Carmen Aguilar, and I'm an associate scientist here at the School of Freshwater Sciences. And I'm very interested in phytoplankton ecology and food web interactions, which is one of the things that we're going to talk to you today. I hope that some of our teachers that we have been working in the past are here with us, and also maybe one of the professors in Mexico. Buenas noches. Hopefully he made it to join us tonight.
And I'm Dr. Cuhel, and I'm excited to be here too. I've been a moderator for NOSB for years. I'm an oceanographer. I have 2,000 days at sea. I love going out. And my work is interested in how rocks, light, and water are interacted with by organisms to make the food of the earth.
And we're very excited to be hosting the Nationals this year, so we hope to see you next April.
So thank you, Melissa, and don't forget, those of you who are watching, that you can't win the door prize if you don't come back next week.
And there'll be a quiz as well.
Okay, so just like the oceans, all the Great Lakes are not the same. And this is a map of the 5 Great Lakes showing from satellite calculations the surface temperature of the lakes during late June. And what you can see is that blue, which is cold, is a common color in northern deep Lake Superior, and red and purple, which is hot, is a common color in the shallow southernmost Lake Erie. 4 of the 5 lakes look similar within themselves, but Lake Michigan, which spans over 4 degrees of latitude, that's more than 250 miles, is able to be warmer in the southern part and cooler in the northern part. And then in the little finger on the left, there is a red area that's Green Bay that's very shallow. So at any one time among the lakes, a huge variety of thermal and other kinds of habitats are available. We can look at some of the characteristics of the 5 Great Lakes to see how they're different, and we're only going to focus on 2. The maximum depth, which is how deep it is, ranges from over 1,300 feet or 400 meters in Lake Superior to as little as 210 feet or 64 meters in Lake Erie. That's a huge difference, and it has a lot to do with the physics of the lakes and how each one behaves when there's a storm or when it's winter or summer. The other part is we call retention time or residence time, which is how long it would take to completely replace the water that's in the lake with water coming in. And that takes as long as 191 years for Lake Superior, which is the largest volume of all the lakes, to as little as 2.6 years for Lake Erie, which is the smallest one and shallow. Lake Michigan is around 100 years, and that means that when you put something in it that doesn't do anything except be dissolved in the water, that about half of it will go away in 100 years, and another half in another 100 years, and so the persistence of materials is great.
A lot of the things that we're going to be telling you tonight are dealing with what kind of things happen in these lakes. And Lake Michigan, which is our main body of study that we have been doing a time series analysis on, and a lot of work on phytoplankton, zooplankton, and some of the things that we're going to be telling you tonight. We're going to be asking you to be thinking about the other lakes here and there but just bear with us. And also we want to talk to you about what do we find in the ocean and why these big lakes are similar in that respect. We're going to be talking about the plankton, and right now I'm just going to tell you that they are comprised of different groups of organisms that would be the base of that aquatic food web. Here in this picture, what I did is I took a picture, and it's not too clear in some things, but you can see the antenna of one of the big zooplanktons. So you can see for scale, this would be some of the phytoplankton that we observe. This is a Fragillaria, we have a Steronella and Tabellaria, and those are some of the organisms that would be part of the plankton, and specifically the phytoplankton. So these are not defined by the size or the taxonomy, but they are carried by the water motion. And sometimes, the zooplankton would be hunting them, as if you will. Now, we're talking about these microscopic organisms. Like I was just saying, they float freely in the water currents of lakes, oceans, and other bodies of water, impoundments, and such. And this one is again showing you some of those organisms that if you see them here, they can get really, really, really long, and some of them are just very small as well. And the word plankton comes from the Greek planktos, which means drifting. There are a lot of different types of phytoplankton and plankton, and we're going to be telling you a little bit about that. Now, most of these organisms are microscopic, and they include a wide variety of organisms, like I was just showing you, some algae, some bacteria, and some single-celled animals. And there are some of the ones that can become larger when they have a planktonic stage. And we're going to be talking to you a little bit more about that on the next session. And so mussels would have this stage that is called a veliger, and they would also be in that planktonic stage. Now, we can, like I was alluding to, we can divide them into phytoplankton, zooplankton, and then bacteria. So the phytoplankton would be your photosynthetic organisms, the primary producers, and require light and nutrients to grow. So you would find them in the upper water column of a lake or the ocean, and they would be your autotrophs, and they would be comprised of all the photosynthetic organisms, including algae and cyanobacteria that used to be considered blue green algae. Now we have our zooplankton in that part, and these are being the animals of that component. The phytoplankton would be your plants, and they would be your heterotrophic consumers. So they would be kind of looking for those types of food in the water. Now the bacteria are very small organisms, and they are also found to decompose organic matter into the basic constituents. So now if you were to think about if you have a phytoplankton and a zooplankton and bacteria, a lot of people have been saying, well, when they are doing photosynthesis, they have cells that might be leaking some of these nutrients, and the same thing if zooplankton eat kind of messy, they're messy eaters, then you would have some of those nutrients also available in the water. Now, as I was mentioning before, we were talking about that these organisms definitely require light. And if you look at the paper that we asked you to look at and read, we have part of the column behind us, and you can see that we would have the phytoplankton being on this area on the top, and they use chlorophyll to convert energy from the sunlight, and they use the dissolved CO2 into carbohydrates. So that's the way that they would be fixing that CO2. Now the phytoplankton there would be in the euphotic zone, which is the part of the water where they would have the best light available to them. And now if we think about also in the past and what we have right now, the phytoplankton are organisms that have produced a lot of the oxygen in the Earth's atmosphere. And so if we think about back then, when these organisms used to start thriving, we have the stromatolites and we have organisms that started giving oxygen into the atmosphere. So they were very critical since the very beginning, a long time ago. So, and we have them still here, and they keep on producing a lot of this oxygen that is required for us to breathe and thrive. So I was mentioning that there are several groups of this phytoplankton, and this is important to think about when you look at, well, phytoplankton, kind of we're telling you a lot about different types of things. So we have very important components of that. So one of them is these diatoms, and the diatoms are organisms that love to be in glass houses. So the outer part of their cell is coated by silica, and that's a very important component for the biogeochemistry of that element. And then we have the golden browed algae, and we're going to show you some examples of that, the chrysophytes, and the green algae, the chlorophytes, blue-greens that we were mentioning before, that are the cyanobacteria or cyanophytes, dinoflagellates, pyrophytes, the cryptomonads, and the microflagellates that you probably all have experience in your classroom looking at some of the protozoans. And when you look at the different types of organisms, you probably go to get some pond water and you will find some of these organisms as well. So I bet you have been one with those. Now let me show you just really quickly some of those organisms that we have been talking about. And one of the things that I want you to think about is the type of structure and the physics of the different algae that I'm going to show you. And then later, maybe on the next webinar, we'll tell you a little bit about what does that mean for sinking and where would these organisms go. So here we have an Acerionella. So you see how they have these arms here. We see Fragillaria that has a lot of the cells one after the other. So you can, if you separate one of these, they would be kind of thin. This one is Cyclotella. And then you can see the chlorophyll, the chloroplast in here. And this is Tabularia. So you see that all of them have different structure, and that's going to be really important when we discuss of where are they in the water column and what happens to them when they are not adrift as much. So we're looking at cyanobacteria, and we were talking about how these organisms are really important in the oxygen evolution of the earth, and in particular cyanobacteria were the organisms that way back in history started giving that oxygen into the atmosphere. Now, these organisms also have heterocysts that are structures that are designed specifically to fix nitrogen, which is from the atmosphere, and that's a really important trait of this group. Then we have little ones that are here in a big aggregate. These are microcystis, and these are ones that we have seen that can be very toxic in some of the lakes and reservoirs. And so sometimes people just call them really nauseous algae, but those are one of my favorite. Then here we have big filaments like Lyngbia, and then smaller filaments like Oscillatoria. So if you look in your pond water again, and some of the waters that you look at, you will find a lot of these organisms. Now, these cyanobacteria, and the reason we're kind of focusing a little bit on those is because they can also be deeper in the lakes. And if you look at the figure that we were showing you, this red band over here was to depict some of these cyanobacteria. And as you can see here, they live deeper, and they take that green light. The red light. So they have the red pigment called phycoerythrin, and they are really good at absorbing deeper in the water. So they can really outcompete some of the other counterparts that they have that they cannot be deeper. So the diatoms don't really like being deep in the water. They like being more close to the surface. And the other thing that I want to show you here, and then that's going to become really important next week, is these unicellular cyanobacteria. And we're going to see how our model has changed after some of these ecosystem engineering. Now, as you can see here, the pigment is excellent at absorbing that green light. And in the microscope here, you can see how they glow red when the green light shines on them. So that's going to be an important thing for them to be in the deeper area. So it's like when you have a plant at home and you have it in the shade and they look usually reddish or darker green. So that's one of those analogies that we can do that. Now, the other ones that look like little trees, these are chrysophytes, and this is Dinobrion, and this is one that we started to see more and more in Lake Michigan. So I'm looking into what really triggers these organisms to be kind of growing fast. Now we have the chlorophytes, we have Pediastrum, we have Cenedesmus that are also very important in Lake Michigan. Dinoflagellates that also have, an outer armor, and this is Serratium. And then they have a flagellate, a flagellum that allows them to move. And these ones have two flagella here that you cannot really see, but that's how they move. And then Russell is going to be telling us about some of these organisms that would be, eating the phytoplankton.
But first, Carmen, let me ask a question.
Oh.
Are there any of the groups of algae that you talked about just now that are more or less common in ocean systems than in lake systems?
Certainly. Some of these organisms that we find, diatoms are definitely very abundant in the ocean. And for example, that Serratium, there's a counterpart that we find there, and some of the dinoflagellates, and that would be a really important component in the oceans.
And another thing that we consider is that the toxic blue-green algae in freshwater are uncommon or unknown in the oceans, but there are very toxic dinoflagellates and diatoms that cause toxic algal blooms in marine systems. So they both have similar concepts, but they're slightly different. Zooplankton are going to turn out to be that way also. Zoo is where we go to see animals, so zooplankton must be animal plankton. And they are typically nearly neutrally buoyant, often weakly swimming. They can swim, but when the current comes along, they can swim within it, but not against it. They can't go any great distance without relying on the water movement. There are all kinds of zooplankton. The herbivores, which eat the algae, the phytoplankton, are like the rabbits in a field. They are small animals that convert plant or algal tissue into consumer tissue. There are secondary consumers, which we might call carnivores, which eat other animals, and they might eat small animals, or they might eat the baby stages of their own type, or they may be omnivores, which means that they can eat anything, large phytoplankton or other animals. We break the zooplankton into several groups, and the two big ones that we're going to talk about today are the macro and the micro. The macro, or large zooplankton, are invertebrates that measure 1 to 2 millimeters long, maybe they're less, maybe half a millimeter, big enough to be caught by a net and seen with the naked eye. Copepods and cladocerans like Daphnia, the water flea, are in this group in freshwater. They can swim. Copepods also in the ocean. It's a large percent, these two, of all the zooplankton. And zooplankton, because they are large enough to be seen, are actually an important food for forage species and the larval stages of many fish. So zooplankton, especially MicroZooplankton, are very central in the food web. Here we have some copepods. Copepods typically have a large antenna that they actually use for swimming. And here is a copepod, another shadow picture of copepod, and they are very abundant. Copepods are very common in lakes in the winter, and they persist through the summer, but the cladocerans become more important in the summer and are almost never seen in the winter. Cladocerans are referred to as water fleas sometimes. Daphnia is one, bosmina. They are diverse and abundant. They exist, but they are rare in the ocean. So cladocerans are a form of zooplankton which is most commonly associated with freshwater. Then finally, not finally, but finally for today, we're talking about MicroZooplankton. Micro meaning small. The MicroZooplankton are not visible when you hold up a drop of water and look at it. They are 100 microns or less usually. They include protozoa, ciliates, rotifers. Here, two different rotifers are shown where the mouth of the one on top is about the same size as one of the Chlorococcus type algae. They eat bacteria. They eat smaller algae and sometimes small animals like protozoa. They are in turn eaten by macrozooplankton. They're usually just barely big enough to be seen or not. Fish don't eat them as much as the macrozooplankton. We sample zooplankton and phytoplankton with nets. On the left here you see a long zooplankton net with a coarse mesh, and on the right here you see the bottle that has the sample in it. And here we're going to just tow a zooplankton net through the water. This is at Can Reef near here. There you see the hydro wire, and then here comes the mouth of the net, and the net, and then the cut end, which is where the animals are collected. Very simple, but difficult to do correctly, and there are many scientists who have spent their whole lives studying zooplankton.
Whoa, that is loaded. That's probably mostly Daphnia.
So that's 20 seconds on the boat.
So one of the things that I was gonna mention is that when we look at these, I don't know if you guys have had the opportunity of looking at zooplankton, but when you look at the sample that comes up and then you look at them, and why Russell is so excited about the cladocerans is that if you have, if you have a copepod, they would swim like this. So you would see them kind of jumping up. Now, if you see a Daphnia, they kind of push themselves back. So then you can see by the way they swim, and some don't necessarily see them, but you can see how their bodies go up and down or kind of backwards. So that's a really cool thing to do when you find these things. Now we're gonna go down all the way into the mud, and in some places the dark areas of lakes and reservoirs. And of course, this is very different than what we would find in the ocean, because we're talking about very different depths, unless you're talking about the shallow areas close to the coast. So now, the benthos occurs, obviously, there's all the way on the bottom. So when we have inshore, we have the non-mud shallows, and we have a lot of sand. And well, you see sand, so it's all very nice and non-compacted. So we have some isopods there, some crayfish, and large amphipods. And then we have the real deep offshore mud habitat. So we have the real mud that can get compacted. And here we find organisms such as mysis, relicta that we see over here. And the reason why I kind of wanted to show you a good picture of it, if you look close, and I don't know, you may want to look in one of the pictures or just type mysis, and these are the opossum shrimp, but these guys have really nice complex eyes. And so they can see a lot, and they are almost like your shrimp. So if you have seen the shrimp, Those are really good beady eyes that they have. Ah, Russell, thank you. So you can see how the eye is very important, and you can tell by that that they're detritivores, and so they kind of really eat a lot of those yummy things that they find in the mud. So now on the left here we have the Diaporia. Oh, my pointer went backwards. Here it is. And they're called scuds. And those are really important organisms for fish. They are like, really nice and nutritious food for them. And then we have a lot of worms that can be segmented and non-segmented. Like we count some of the worms, and usually we have some of the really good numbers in the summer. Now, also present in the coastal areas, we have Clarafora, and probably a lot of you, if you are from this area, have dealt with the odor of Clarafora that brings a lot of fish and a lot of other organisms that really stink when they are rotten on the beach. So that's another big component that we have now in Lake Michigan shore, that sometimes we have really good years for Clarafora, and people are very unhappy about that because they really smell. And that's what happens, is that they're really long strands of filamentous algae. And if we were to have one of our rocks that we collect, mussels and such, when we bring it up, this Clarafora would be kind of going all the way to the bottom when they collapse in there. But if you look at them underwater, they kind of sway back and forth, and they get to be quite big. Now, the diatoms that we find there would be called benthic diatoms, and they move along the bottom of surfaces. So they kind of become kind of slimy, almost like snails, and then they move around that way. And they can also be attached to the Clarafora. Then the other organisms that we find there are snails and different insect larvae. The snails also can attach to the Clarafora, and we have a movie, but I don't know if that's the one where we saw a lot of little white specks on the Clarafora, and we thought, oh, those are oxygen bubbles, but it turns out that there were a lot of snails there. So they can be very abundant. Now, how do we sample the benthos? Here we have a ponar that is named after the initials of 5 people that designed this. So basically, there's big claws that go all the way to the bottom, they close, and then we bring a sample that then we can sieve and count everything, and it's a quantitative measure of that parcel of the bottom. The other way that we sample is by using a remotely operated vehicle, and this is one of our favorite ones that we use here at the School of Freshwater Sciences. Now, this movie is to show you the ponar in action. So it's going down, and you can see we have a ponar cam that we call, and you can see what you are going to be sampling. So down there we see a lot of organisms, and it's going to go down, and see then it creates a little resuspension of material. Then when you bring it up, starts closing on itself, and then it closes completely. There you go. And then you bring it up, and you have your sample there, and leaving behind a bare spot. So now you bring it onto the ship, and you have your sample here, and we are able to then sieve, and a lot of our students have helped us count a lot of those samples, measure and count all the things that we bring from our ponar. And in some of these cruises, it's a lot of animals to count. Now here we're going to show you an ROV slurp from a rock. So the thing about collecting with the ponar and collecting with our ROV, you can see that although we have the ponar cam, sometimes it would be difficult for us to be sampling on top of the rocks. So here with the slurp gun, we can measure an area with, there's 2 lasers that allow us to say, well, this is the distance between these 2, and we can sample like that, and we can be more the way that we sample, we say, well, we'll sample the top of the rock or the bottom of the rock and such. So that allows us to be more specific on the things that we are sampling. One of the other things that we have there, and I know that some of you that have worked in some of those areas that have sometimes less oxygen, you can see those chironomids, and they look red, and a lot of kids go, ew, they look gross, because they look really red and kind of, hard. But those are organisms that live on or in the bottom habitats as part of these benthos that we have been talking about. And they're detritivores. They eat a lot of the materials and organic materials that are on the bottom, and phytoplankton that would sink, or they would sift through the dirt for some of the really nice nutrients that they're looking for. And some organisms like this would be favorite food of the bottom feeding fish. So now we're looking at what things would be eating the phytoplankton and what things would fish be eating. So some of these fish would be eating our zooplankton, some of these would be eating our chironomids. And now we're going to be showing you a scene from the bottom. And this is one favorite of ours, and we called it Khufu Fighting. And you'll see why. I didn't realize that the little crayfish could have those kind of moves. And look at those white specks on the Clarafora. That little luscious hair is Clarafora. And those are the ones I was telling you that were snails and not bubbles of oxygen. So look at that guy. He was fighting us and fighting us. And in the end, you'll see that he makes himself big. And when you think about a predator that is not going for it right away, they will not have a chance with this guy kind of waving the arms, up in the water and not trying to, trying to escape being collected in this case. So, so those are one of the things. So you notice the Clarafora, you notice the crayfish, a lot of those snails, and a lot of the rocks in there. And we're going to be discussing what other organisms we have been finding along with that Clarafora, and that's going to be some of the themes that we're going to be talking next week. So now Russell is going to be telling us about the fish. But then if you look at your graph that we showed you before, we have some of the fish over here with the zooplankton and phytoplankton, and some of the fish closer to the bottom.
That's very good, Carmen. One of the things that we're going to be talking a lot about, and especially at the Ocean Bowl, is interactions between, for example, physics, biology, and chemistry. And the benthos is an interesting zone because the physics of stratification has a lot to do with the way that benthic organisms make a living. Right now, though, we're going to talk about fish because that's the next step in our food chain, which is not really a chain, it's more of a web, as we'll discuss in a little while. Fish are vertebrates, which distinguishes them from previously discussed animals. Everything before was either a photosynthetic organism or an invertebrate. Fish are vertebrates. The forage fish are smaller. They are often planktivorous fish. That means that they eat plankton that are themselves eaten by higher order carnivores. And then piscivorous fish are fish that eat other fish. A piscivore is a fish eater. Usually in the forage fish group, that is the food that they eat, is usually smaller forage fish. And large piscivores can eat nearly any fish. A big lake trout, for example, can eat almost 100 smaller cutthroat trout in Yellowstone Lake in a single year. In the forage fish, both humans and other animals make use of forage fish for food. Of course, the forage fish don't see themselves that way. But they convert the plankton biomass into something which is accessible to the higher food web. The top picture there is a chub, and we used to eat a lot of smoked chubs from Lake Michigan. It was a very common commercial fishery, and smoked chubs were pretty tasty. Unfortunately, chubs have disappeared from Lake Michigan largely, and that's one of the things that we'll be talking about next week after you do your homework. Down below the chub is the alewife, and alewife and smelt and chubs and minnows are really great food for salmon and perch and a variety of larger fish that are not necessarily, some of them are top predators and some are not. So perch, for example, eat them, and then perch are kind of a middle predator. Here we're going to look at alewife from the ROV while we cruise along on a mini drumlin. And did I click it? Maybe I didn't click
No,
it.
you have to click it.
There we go. Okay. And here we're in this is in 2003, and right away see the school of fish. Okay, and that school of alewife is in a coastal zone right near this underwater ridge, a little clay drumlin. For those of you who are in marine habitats, the alewife would be similar to menhaden, any kind of estuarine killifish, a small fish that eats plankton, that has schooling behavior where a bunch of them swim together, and that is planktivorous and usually the prey of other fish. There's a few more alewives there, and then you see the bare clay and the rocks. And so this is one example of using a survey instrument like a remotely operated vehicle to look at the habitat and the ecology of the habitat kind of visually, and then use the slurp gun that you see on the top of the screen to slurp up samples of the bottom habitat for our studies. Then there's the carnivorous fish. These are fish that eat animals. Okay, so an herbivorous fish or a planktivorous fish eats both animals and algal plankton. Carnivorous fish actually directly go for large invertebrates and other small fish. They don't really eat any of the photosynthetic organisms on purpose. They can hunt, they can stalk, they can chase down prey. Perch, whitefish, and sculpin. Perch and sculpin shown at the bottom are predatory fish that are carnivorous, but they're not strictly piscivorous. They'll eat a worm, they'll eat an amphipod, anything they can get. Piscivorous fish are near the top of the food web, and they are, for example, the large lake trout. In Lake Michigan, for thousands of years, lake trout was the top predator. And it was the animal that kept all the forage fish under control in terms of populations. It was the best known indigenous top carnivore in Lake Michigan and a very popular fishing item. Other ones like walleye, we know walleye pike is one that is very popular with sport fishermen. And then a lesser known but still pretty big, physically big fish that's common in Lake Michigan is the freshwater cod, or burbot, which is a bottom-dwelling piscivore that lives in the rocks on the bottom. So here, for example, we're going to look at lake trout during the dark era. This is in deep water off near the middle of Lake Michigan, and it's a pretty dark time. Here comes a lake trout cruising along. They live individually except during mating period seasons, and they cruise around looking for food. Here is at that same time of the dark period at 52 meters or about 175 feet, and there on the bottom you see a nice burbot. He'll take off. There he is moving around in his little home canyon area waiting for a sculpin or two to stop by and be a meal. So we've looked at a variety of photosynthetic or primary producers and things that eat them and the things that eat them. And although we have barely touched upon it, we really have kind of begun to develop what we'll call a food web model. And this one here is a drawing from the paper that you should be reading that represents a square area, a square meter of deep part of the Lake Michigan water column. And what you see is the phytoplankton, which are in two colors. Up at the surface, there's green phytoplankton, and they are able to live there because the light that comes in is white, and they can absorb the blue and red components of the light and use that for growth. Then down deeper underneath them, there's a gold zone, and that's the zone of the juicy steaks or the bean burritos of the food web, the diatoms that Carmen was talking about. And they are golden colored algae, and the reason why they can live there is because their pigment called carotenoids, which notice the carrot color of the zone, is able to absorb a little bit of the non-blue light, not the green exactly, but they can make a good living in the middle water. And then below that are a few algae that are really well adapted to very low light. And so that's our primary producers. That's the food of the food web. And then when it gets calm, or they, under circumstances where they might, what we'll call die, they will sink to the bottom, and the little circles dropping down to the bottom are algae and the poop of things that eat algae that are sinking down to the bottom. Then you see the copepods and the cladocerans are the animals that are swimming mostly in the carrot-colored or golden zone of high nutrition algae, but also some in the surface. And they come up to that zone and they feed on the algae that grow in the upper water column. Then you also see some schools of forage fish, little collections of alewife, or could be any forage fish, smelt, and they are up there to eat on the zooplankton. Down at the bottom, you'll see the few amphipods and a mysid or two that are the benthic detritivores that feed on the rain of organic material that sinks to the bottom. And so this is our what we'll call our basic food web. And this kind of food web with different scales occurs in almost every aquatic habitat, from a drainage ditch to the middle of the Atlantic Ocean.
So one of the things that I just wanted to mention before we move along is that, okay, so when you saw that we were sampling with the DePoner, and so the DePoner goes down and whack, we get part of the amphipods and the mysids and the stuff that is down here, and that could be the mud or it could be the sand. So that comes through the water column. So then Russell said that we would sample zooplankton and phytoplankton with different nets. So what does that mean? That the big net would then go, for example, we go all the way to the bottom, so not to drag it on the bottom, because that would not be good. So if you get a mysid in your net, that's kind of not cool if they're on the bottom.
Except at night.
Except at night. Well, that's another story. So your net would be, so we lower it down and then we bring it up as fast as we can, because as you know, the zooplankton, as I was telling you, they would be trying to escape. So we grab those guys. So they would be with bigger pores, so we can get the bigger animals, in this case, the zooplankton. So then we send another net down, and then this one, we don't go all the way to the bottom, except for right now that all the water is all mixed. But so we would go to the bottom, and then we would bring it up again, and then we would get with the smaller mesh some of those phytoplankton. So this is kind of like in our virtual water column, we can sample all kinds of things. And if we were interested in the fish, like John Jansen is, he would then sample specifically for those organisms. Now, the other part that Russell is going to tell you about is the birds.
Right, and we're just going to briefly mention birds. In aquatic systems in freshwater, particularly where the water is shallow, birds, which are also vertebrates, are a considerable predator for some aquatic organisms. Some, like ducks, for example, there are many kinds of ducks, and some ducks are herbivorous, and they go down and they feed on, for example, the Clarafora, and they rip it out and they eat that. Other ducks, like mergansers, for example, are carnivorous, and they go down and they're looking for mussels and worms and amphipods and small fish. And then, of course, seagulls and cormorants are really vigorous, predatory birds that dive into the water to catch fish. Seagulls commonly eat forage fish like alewife, especially in the summertime, and cormorants can swim deep underwater very quickly, and so they can chase and capture even perch. They eat quite a few perch.
So now, you probably all have seen the kinds of food webs or food chains and a big discussion about that. So, you have to just think about what do you have in your system or what do you want to talk to your students about to talk about these kind of issues. So a lot of even now we have a lot of these diagrams that shows you the relationship between the different types of aquatic organisms, and they connect with these and the bottom and such. But once you really start going and trying to dissect all the different compartments, it becomes really like you want to pull your hair and go, this is like way too much. But if you really look about the big ticket items, so to speak, you will find that you'll have your microinvertebrates, and you have a consumer here, you have resuspended material that other organisms can eat, and then here you have like the direct line, right? The phytoplankton, the zooplankton, the consumers, and the secondary consumers. Well, this looks really good, but in reality it gets really messy. So so this is part of would be your typical food chain. But then these guys could eat some other things, and then these guys could be peeing, and these guys could be eating some other things. So just be aware that there's bacteria, those damn bacteria that keep on coming back and back, and you have the protozoans, and you have all these other organisms that put a wrench in your system. So always be aware of when you're talking about this, how to do the connections and what to talk about. Now, this is a more realistic food web, and it's still simplified because we don't have the bacteria, we don't have some of those smaller organisms, but you can see the different kinds of fish. And then we're going to be talking about these other ones a little bit next week. And so we're going to be talking about, we talked about these, we talked about those, we have seen those. So you will see the usual suspects here. And now Russell is going to be telling us a little bit about the other component of this paper that we are really interested in. Now we kind of created this idea of what lives in the bottom, what lives in the middle of the water, what lives on the top, what things are interacting. And now we're going to put another wrench in the system, which is what happens when invasives come into your reservoir, lake, even the Great Lakes, as we talk about. There's all different ones, but some of them gets more of the other for some organisms and other ones. And we're going to mention that next week. So Russell is going to be telling us about some of those invasives that are putting a wrench in the system.
That's right, Carmen. We're going to now use our knowledge of how the food web works. Of course, we kind of zoomed through it, but you've studied this in and out for months and months. We're going to use our knowledge of how the food web works to try to predict what would happen when we had a large invasion by some external organism. And that's a part of our homework is going to be this. So here we're going to have a couple of tables in which we look at what year was this organism first found to be successfully living in Lake Michigan. What is the family that it's in, the common name, the scientific name, and then over on the far right I've put type. And in type I'm thinking about what is its role or its activity in the food web so that we can think about how it might affect our model food web if it came into Lake Michigan. And some of the ones that we're going to be focusing on in the paper are identified with these little arrows here. For example, Alewife. Many of these are very important, but we're going to focus on some of them next week. Right now, we're going to say that in the 1870s and into the early and mid 1900s, there were a number of fish that, of course, 0 is a number, so there were a few fish that made their way into Lake Michigan and became established. Some of them were brought here intentionally. Some of them swam up the St. Lawrence Seaway and up through the Erie Canal and through the Welland Canal, which was the way that got materials around Niagara Falls, which was originally a nice barrier to invasion. So Chinook salmon, for example, which is a piscivore. What does that mean? That means it eats fish. Then rainbow smelt, which is a forage fish. It's a planktivore, very similar to alewife, which is going to come in later. That came in the early 1900s. Common carp, the bottom-dwelling golden carp, which is, we'll call it a middle kind of fish because it will eat any detritus and plants and stuff off the bottom, but it will also eat snails and crayfish if it can get them. Alewife, which is a really important planktivore that is a forage fish. And then we have a series of salmon that were either imported or introduced. White perch, which is very common in Green Bay, the round goby, and the Eurasian ruff are very recent fish invaders that may have significant effects on the food web. We're really going to focus a little bit tomorrow or next week on the sea lamprey, which is a parasite of fish. And then here we have some microorganisms that are kidney disease, whirling disease, and a viral blood disease that are very bad. Invasive mollusks include, of course, many of us are familiar with the zebra mussel and the quagga mussel. And then also we have a variety of snails and clams which have been known to come in. Carmen is holding up a bag of Pisidium. Actually, no, that's a spherium. It's the fingernail clam. And those we collected in a ponar grab. You need to pull it away from, pull it away a little bit. Yeah, up. Perfect. Okay, the fingernail clam, which is becoming more and more common in sediments of Lake Michigan.
So these are tiny, 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 tiny shells. So if we wanted to count these, we would be pulling our hair. So we weigh them.
So we counted 2,000 of them and weigh it, and then we weigh how much there is and then calculate. There are a number of invertebrate non-mollusks, including especially the spiny water flea and the fishhook water flea are two that have been in the lakes for a while that were originally feared to have major impacts on the aquatic ecosystem, and we'll look into that. And then the new one that just came in 2007, the blood red shrimp or the red mysid, blood mysid, is also one that is recently invading. And again, some of these are carnivorous, some of them are herbivorous, some of them are detritivorous, that means they eat dirt, and then some of them are omnivorous, which eat anything that they can get. And so we'll be asking you to think about this. And the invasive microorganisms, we have a few. Staphanodiscus, a centric diatom, and a red alga. And one of the questions we'll ask you for homework is, how come the list of invasive microorganisms is so short compared to, say, fish or invertebrates?
Go ahead.
Oh, okay. So in the invasives, we're just going to quickly look at a couple of them. In the invasives of the zooplankton, we have the fishhook water flea, Cercopagus pangoi, which is a zooplankton predator. It eats other zooplankton, and it had the potential to really seriously change the zooplankton part of the food web. Another one that's very similar is also a cladoceran, a predatory cladoceran, called Bithotriphis, or the spiny water flea, as opposed to the fishhook water flea. And it also is very large, and it eats other zooplankton. This compares the size of the two of them with the larger Bithotriphis down below, and on its back it has some babies that it will be releasing into the water. And so the benthos, there are three really big invaders. Hemimysis anomala is the blood red shrimp, and we have zebra mussels and quagga mussels.
So here we have the the blood red shrimp, the Hemimysis anomala, and you know, if you were to lift one of these rocks that I'm just going to show you here, you see that there's the water line where is the dark and the light, and where we find these organisms would be under the rocks, always tucked under. So I remember that we brought a rock like that, and we just kind of were filming them, and we didn't count them because that's not the type of organisms that we work with, but there were a lot of them when they started invading. So they can really be detrimental to our system, and they can grow in huge numbers. So this was found in 2006, and it's a small shrimp. And it's also native from the Black Sea, and we're going to talk about that some of our invaders are coming from that area in Europe. Now, one of the ones that you probably all have seen is the invasive zebra mussel. And of course, they would attach to any surface. They are Ponto-Caspian, the Caspian Sea. And so a lot of these came through the ballast water in ships. And so here we're showing you on the left how they encrust everything. And Jim Lubner took this picture some years ago when they brought this card up. It was completely encrusted with the zebra mussels, and unfortunately the shopper was never found. Now if you look on the right, you'll see that the zebra mussels were really restricted to shallow, warm waters of Lake Michigan. So you can see if you hug here the coast, this is where we would find them. And that's going to become really important when we discuss what happened later in this invasion. So just keep that in mind, and we're going to probably start with this next week.
So this little movie in here that shows, that's in a graph, the graph shows, well, the movie shows a rock covered with zebra mussels in Green Bay that is sucking water up, removing all the plankton from it, and then spitting out the plankton that they don't want to digest. And during the late zebra mussel period, we found that this was a very common activity on the bottom, but there was a new animal that was coming in and starting to make a good population, even though the zebra mussels were very well established. So we have developed really the barest concept of an aquatic food web. But we did try to get across the idea that there are zooplankton that eat other zooplankton, and there are even algae that eat other algae, if you can imagine that. We have identified some of the successful invasive species and their nutritional habits in the type category. Your homework for next week is to determine how the listed invasives might alter our model of water column communities. And then next week, we're going to talk about the game changers and the biggest ecosystem engineer of all.
So thank you very much, and we're ready for some of your great questions.
Okay, well, thank you very much, Carmen and Russell. I actually think that this topic is going to be of a large interest to the NOSB students who are competing. Food webs are one of the questions that they get a lot during the competition, and as we know, as you said, they can be very simplified, but that doesn't necessarily show exactly what's happening in the actual water column and the habitat. So thank you again, and it looks like we have a few questions that came in from earlier on, so all of you who are still on, feel free to type in any questions that you may have had from any part of the presentation, or if you even have questions maybe about the paper that they're going to be discussing next week.
Also, I'd like to put in a request, and that is because we're talking again next week, we'd like to know if the level that we approached this at was good for you. Or do you know all this and you want us to go deeper? So please feel free to give us constructive criticism.
One other thing you'll notice is on our website, the Professional Development Webinar Series link on our website, you can type in comments under each of the speakers' names. So if in the next week you have questions that you think of, feel free to type them in there, and we'll either try to email you back with the answer, or we may try to answer some of those questions next week during the presentation. Let's see, I have one coach that I know is asking, yes, please go deeper and more in depth. Most of this stuff we know as science teachers. So there's one comment for you to go a little bit more in depth. One of the questions that we got early on was saying that there were episodes of, I think it's toxic phytoplankton blooms that are appearing along the world's coast. Is this also happening in the Great Lakes as well?
What we seen in the coast and the ocean, we have what is the name of that diatom?
I know the name of the toxin, domoic acid.
Yeah, so there's a diatom that the name Nischia.
Yeah, there's a Nischia.
So they release a lot of domoic acid that is a big problem, especially in the Pacific Ocean. And we have, for example, not in Lake Michigan, not in Lake Superior, and Lake Erie has an issue with the microcystis and anabina that can be very toxic. So if you look at Lake Erie, and we were talking about some of these temperatures, and because it's very shallow and it's a different orientation, that really makes it be mixed more often. And they have a lot of the issues that a lot of the areas in the, well, not a lot, but some of the areas in the ocean have, which is also the prevalence of low oxygen zones. So Lake Erie would be one that is very susceptible to having those issues with toxic cyanobacteria, not the diatoms, but the cyanos, and also those areas of the very low oxygen concentrations that they would see mainly in these areas. Do you want to add something?
The microcystis, which is the toxic, is one of the cyanobacteria that makes the toxin microcystin. The microcystin is a cyclic polypeptide. It's about 10 amino acids linked together into a circular molecule, and it attacks the liver. So organisms that drink water get liver poisoning. And the most common thing that happens is that, for example, dogs and cows and other animals will run down to the edge of a small lake, or a big lake like Lake Winnebago, and they will drink the water, and the water will contain the microcystis, and then they will become sick or die from that. An interesting related thing was that in one case in the jungle where people were using lake water to dialyze for people that had kidney problems, is that people were dying from dialysis, and it turned out that the water that they were dialyzing against was loaded with microcystin from toxic cyanobacterial blooms.
Wow.
We don't have to see
We,
it, so.
okay. We have another question that was related to blooms. In temperate marine areas, the plankton bloom seasonally in the spring, and the smaller ones typically in the early fall. Are there similar seasonal blooms in the Great Lakes, and are they typically nutrient limited?
Well, there are. Seasonality in the Great Lakes, in all of the Great Lakes, is a common feature. Each of the Great Lakes behaves completely differently. When you read the article that we are assigning for next week, you'll see that Lake Michigan has changed, but up until a little while ago, in April or so, late April, there would be a very strong bloom of primarily diatoms. And then as soon as the water started to warm up, they would sink out and grow deep in the water. Then late in the season, in the fall, early fall, there would be another bloom as the turbulence of fall storms and the cooling of the lake would enable the plankton to be mixed up to near the light and would also mix nutrients from the bottom. So the seasonality has to do largely with light and nutrients. In the spring, the days start getting longer and longer. After March 22nd, the days start getting to be pretty long, and so the light that's required for the algae to grow is better. Still, it's usually cool and well mixed, and the nutrients are very available.
One of the things that you'll notice, of course, with the weather and the climate change is that you have been, for example, we see extremes now in our area where there's summers, there's winters that we don't have any ice and it's not that cold. And that really has a big impact also on what we see on the season coming forth. So, so that's a really important component of, for example, the effects of the drought and the effects of last year was one of the warmest summers that we had in a long, long time. And when you even looked at the map of El Nino Southern Oscillation, you could see that the Great Lakes were 5 degrees centigrade above normal. And that is a very important thing to also think about.
One of the things that is important in blooms and the food web cycle is that the spring bloom has occurred for millennia, and the animals that eat the spring bloom are ready for it. And so there is a balance between the production of plankton, phytoplankton in the spring, and their consumption by herbivores during their blooming time. One of the things that you can think about, and we'll talk about next week, is if regional climate change results in earlier warm weather, how might there become a disconnect between the phytoplankton spring bloom and the activity of herbivores that are adapted to eat them?
Right.
Okay. We had another question asking if the chironomids are they commonly called bloodworms, or are bloodworms something different?
Nope, those chironomids are bloodworms. And why are they called that? They're called that because they're red in color. But why are they red? They're red because they're loaded with blood with hemoglobin. And why is that? And that is because they live in the mud and they eat mud. And in freshwater, shallow freshwater systems, mud becomes anaerobic without oxygen very rapidly from the bacteria eating the organic matter. So what the bloodworms do is they come up to the surface of the mud, they circulate water through their gills and oxygenate a huge amount of blood in their bodies, and then they dive down into the mud and they feed down there, just like a sperm whale will dive down thousands of feet to find squid. They'll dive down thousands of microns to find good dirt where they can eat even though there's no oxygen.
Okay, thank you. Another question is the food web model you presented very similar species in all the other Great Lakes, or are there significant differences from lake to lake? If there are big differences, what are they?
Okay, the fundamental concept of the square meter food web, or of the one that's on the screen right now, is common among all the lakes. The scale of each component differs, but all the lakes have phytoplankton. All the lakes have zooplankton. All the lakes have some organisms that live on the bottom or in the mud, and all of them have various kinds of fish. It's the balance among them and the continuity of them that differs among lakes. For example, in Lake Michigan, seasonality is extremely high. In the winter, it's cold and the water is well mixed, and in the spring the sun comes out and the water is well mixed and there's good production. But then in the summer, the surface water becomes very warm and the lake becomes highly stratified, and all of the recycling is down at the bottom and the phytoplankton are up at the top. So the summer is a period of relatively low production. In contrast, in Lake Erie, the sediment is so close to the surface that it doesn't take that much wind to mix the nutrients from the compost heap at the bottom into the lit zone up near the top. And so the productivity of Lake Erie is more continuous throughout the year. In fact, it's worse in the winter because it's ice covered, and so there's no turbulence to keep the organisms near the light. So there's kind of a little disconnect in timing, but they all have the same fundamental structure. Do you think that's? Yeah.
And like I
Okay.
said, we also need to think about, for example, in Lake Superior, that is so deep and colder, the type of organisms that, for example, the type of fish that are there are very well adapted to those kind of conditions. And so the other thing that we have been seeing is the fisheries, and we're going to dwell on that a little bit more next week. But the fisheries has really taken a big hit with all these invasive species and how it has changed. So now we are seeing that a lot of those fisheries have been going away from the different lakes, and I think all the lakes have similar issues with respect to that, except for maybe Lake Superior. They still are very oligotrophic.
Okay. Another question was asking, does biodiversity vary from lake to lake across the Great Lakes, or are they also similar?
Absolutely.
Yeah.
Absolutely. There is an ecological kind of mantra that says that biodiversity is highest in the most oligotrophic or low nutrient environments. And so that would say that Lake Superior, now Lake Michigan probably, would have a much higher degree of biodiversity than Lake Erie, for example. And this is largely true. There are a wide variety of organisms, but in rich habitats, certain ones tend to become really dominant because they are well adapted to a high food source. Where in very poor environments like Lake Superior, there are so few things that there's an opportunity to have different lifestyles cohabit. So there is a substantial gradient of biodiversity across the Great Lakes.
Okay, thank you. Those are all the questions that were typed in so far. We have a few more minutes, so if you have any other questions, feel free to type them in right now. Like I said, if not, you can always either email them to NOSB at oceanleadership.org, or you can type them in as a comment on our Professional Development Webinar Series webpage.
And I'd like to ask a question of the observers or of the attendees, and that is, how many of you have looked at our website called Quagga Mussel Invasion? Thank you.
If those who are participating, if you see up at the top, there's a little person that looks like they're waving. You can say set status and you can raise your hand. If you looked at the website, if you raise your hand, I can do a quick count of the participants who have. Let's see, it looks like a few people are putting it in. So it looks like
Michelle. Hi, Michelle.
three or so said yes. Someone's asking, where is the paper to read for next week? If you go to www.nosb.org, in the very top of our website, there's a link you can click on to get to the Professional Development Webinar Series page. And under Russell and Carmen's presentation, there is a link. It says, if you go to participate, please click this link and read the paper. Someone is asking, what kind of classes do you offer for teachers in the summer?
Well, that's going to be interesting. We are trying to develop some classes maybe for next year. And because there's a big interest for teachers to have science classes. And so we're really thinking about that, and maybe with Caroline Joyce, that you guys know as well. We have been toying with this idea, so we'll keep you appraised of what develops, but we're thinking that maybe we'll offer something the summer of 2014.
Okay. Well, we're just about out of time, so unless there are any last-minute questions coming in, I'd really like to thank both Russell and Carmen for presenting tonight. Again, I think this is not only just an interesting topic, but one that obviously comes up a lot in the NOSB competitions. And like I said, I think it's good for everybody to get an idea of food webs and how they work both, either in the ocean or in freshwater environments. I really want to make a plea that for all of you who participated tonight, please join us again next Thursday. We're trying something a little different this year with the webinar series, where we're trying to make this so that there is a bit more professional and personal growth that you can get from participating in these webinars. So this is the idea of having Carmen and Russell present a paper of theirs next week, something that you can read and be able to ask questions about, and maybe get a little bit more in depth into some of the current research that's going on in the Great Lakes. So I do hope that you'll participate next week, and if you know any other coaches who maybe didn't participate tonight, we will have this recording posted on our website tomorrow. So starting by about tomorrow, noon time, check back on our website, and the recording will be there not only for other coaches to watch, any other educator who you think might be interested in it, but of course the teams as well who are participating in NOSB. It'll be a chance for them to study for the competition. So again, I hope that you will all join us again next Thursday, and also share the recording of this webinar, because I think it's both interesting and relevant for the competition this year.
Now, we're going to assume that you have read the paper, which is a nice review that gives a lot of resources for looking at specific topics. And we are going to use numbers from our own collections to test hypotheses about changes in food web structure based on invasive species activities. So we're going to be a little bit more data rich with real live actual data that is used from our actual work to understand how the food web changes in response to a structural change.
Okay. Well, again, thank you, Russell and Carmen, for presenting tonight, and thank you, everyone, for participating. I hope that we will see you all again next Thursday. Just one last note, we will have a presentation next Tuesday night at 8 p.m. as well. We're going to have Peter McAvoy talking about the Great Lakes Compact. So it's actually a really interesting topic about Great Lakes water policy. And obviously, again, this is something that is going to be covered in the competition. So we hope that the coaches will participate both Tuesday night next week and Thursday again for Russell and Carmen's second webinar. So once again, thank you, and I hope you all have a good night.
Thank
See
you.
you next week.